Hello class, meet me again for the soil biology class. Today is lesson 11 and we'll be discussing about mycorrhiza. Some of you may have heard about mycorrhiza because the soil organisms are very close to our lives. But what actually are they? Mycorrhiza comes from the words myco, meaning fungus, and rhiza, meaning roots. Mycorrhiza are symbiotic association essential between fungi and the root of living plants, which is primarily responsible for nutrient acquisition from the soil in exchange for plant carbon. There are four groups of mycorrhiza categorized by their morphological and physiological characteristics, including arbuscular mycorrhiza, ectomycorrhiza, ericoid mycorrhiza, and orchid mycorrhiza. Among them, arbuscular mycorrhiza and ectomycorrhiza are the most commonly found and studied, and those we will discuss more about the two here. Then, based on the evolutionary history, it is reported that the arbuscular mycorrhizal fungi are the earliest to exist in the world. These fungi appeared in the early Devonian era over 400 million years ago and are believed to facilitate terrestrial colonization of ancient plants. On the other hand, the ectomycorrhiza evolved much later, around 50 million years ago. It is reported that 92% of terrestrial plants form mycorrhizal symbiosis, and among them, 71% are arbuscular mycorrhiza and 2% are ectomycorrhiza. Arbuscular mycorrhiza were found in many kinds of plants, including ferns, gymnosperms, and angiosperms, while ectomycorrhiza were found in relatively lower number of plant species. And you can visit the link provided in the slides to find more about it. As for the structure, arbuscular mycorrhiza and ectomycorrhiza were very distinct. Arbuscular mycorrhiza penetrate root cells to form arbuscles and fascicles to facilitate nutrient exchange inside the root, while ectomycorrhizal fungi stay outside, forming a dense fungal set, we call it mantle, on the root surface and a network, or we call it heart net, between root cells, facilitating nutrient exchange without entering the cell. These are several examples of mycorrhizal fungi. The arbuscular mycorrhizal fungi on the left cannot actually be seen with our naked eye due to their tiny size. The fungi from this type, including those from Glomeraceae, Acalosporaceae, Diversisporaceae, and many more. On the other hand, the ectomycorrhiza were bigger in size and sometimes form a fruiting body or mushroom. The examples of them are Tricoloma, Rusula, and etc., and some species of this mycorrhiza are even edible. These maps show the global distribution of mycorrhiza. Red color indicates a higher number of fungi and blue color indicates a lower number. Based on the maps, arbuscular mycorrhiza were more distributed in the tropics and ectomycorrhiza were more distributed in temperate. In the roots or rhizosphere zone, not only do mycorrhizal fungi exist, there are also some bacteria, other types of fungi, mesofauna, and other soil creatures. All of them compete, coexist, and sometimes cooperate, and this needs our understanding and consideration, especially when we want to utilize them for field application. Through the mycorrhizal network in the soil, as well as the other cooperating microbes, plants can send signals to their kin, non-kin, and even different species. This condition, however, will be affected by the plant species and environmental condition, and will result in different interaction or responses between plants, including positive interaction, commensalism, neutralism, and even antagonism. The other fascinating things about mycorrhiza is that these organisms is a carbon reservoir in the soil. As they form symbiosis with most of the terrestrial plants and receive carbon from them, they actually become a carbon storage down in our soil. Carbon sequestration varied among mycorrhizal plant types, with arbuscular mycorrhiza as the highest contributor, followed by ectomycorrhizal and ericoid mycorrhiza. With those roles and benefits, the study of mycorrhizal utilization is expanding, and one of those is for biofertilizer. To utilize mycorrhiza as a biofertilizer, many steps are needed. This process should be started with isolation and identification, in which can use both morphological and molecular methods. Then we should do tests for the isolates in the lab scale, propagate, and finally use them in the field. Several land rehabilitation and reclamation programs were reported to be successful with the application of mycorrhiza as biofertilizer. While mycorrhiza are abundant in the ecosystem, there are threats to their existence, such as soil disturbance, massive chemical use, pollution, climate change, and etc. Thus, we need to take whatever action we can do to overcome it, such as conserving and preserving the habitat, practicing sustainable farming, reducing pollution, and conducting more research about it. And as for me, my research interests focus on mycorrhiza and plant-fungus interaction, particularly on how mycorrhiza symbiosis can establish in disturbed ecosystem and what strategies are posed by its symbiont. Some papers were published, and you can visit my Google Scholar page to see more about it. Here are the list of some high-quality websites for your future reference. This is the end of the session. If you have questions, feel free to contact me by email, and see you in the next session.